Good afternoon, and welcome to our weekly Research in Action series brought to you by the Division of Research at Florida Atlantic University. My name is Jeannie Viviani, and I will be today's moderator. It is my great pleasure to introduce today's speaker. But before we get started, we want to welcome you to ask questions of our speaker using the Q&A feature of the webinar. Please type your questions at any time, and at the end of the speaker's presentation, they will answer as many as they can. Dr. Luiz Campelo dos Santos received his BS in History and MS and Ph.D. in Archeology from federal universities in Brazil. He is now a postdoctoral fellow of the Department of Electrical Engineering and Computer Science at Florida Atlantic University. He joined the research group of Dr. Michael DiGiorgio in October 2021, where he works applying and developing methods in computational biology. Since 2018, Andre has been analyzing the DNA found in archaeological remains, especially in human skeletons, to investigate the ancestry and migratory movements of human groups in the past. Andre's research has been funded in media outlets such as the BBC and Discover Magazine and Gizmodo. Andre, welcome, and the floor is yours.
Thanks, Jenny. Thanks for this introduction. Good afternoon, everyone. So it's my pleasure to be here today to present an important portion of my research, the research that I've been developing here at FAU for the last 1.5 years under the supervision of Dr. Michael DiGiorgio in the Department of Electrical Engineering and Computer Science, the College of Engineering and Computer Science. So my research revolves around migration routes of ancient people. And why do I study ancient migrations? Why is it important? So basically, migration is an important aspect of human history. Our human history has many chapters, and many of those chapters, they were shaped by migrations. People's lives, they are changed forever by migrations, and as such, our whole human history is also shaped by these migrations that happened in the past, be it the recent past or the ancient past. So migrations also gave us hints on how humans adapted to and settled different environments, from deserts to very cold environments like Siberia, very dense forests like Amazonia, and also other environments, different environments such as the highlands in Peru, in the Andes. So humans, they dominated all these different environments. They changed all these different environments. So studying migrations give us hints on how they adapted to these different environments. How long did it take for them to settle these different environments? And how long did it take for them to move from point A to point B? So if we are talking about recent migrations, we can look at historical documents. They will give us a lot of information regarding these recent migrations. But if we are looking at ancient migrations, deep ancestral migrations, historical documents, they are not enough for us to study those events. So as such, there are many gaps in our natural history. When we are talking about the ancient past, the deep past, there are still many gaps. And we study ancient migrations to try to fill out these gaps. And one way to study these ancient migrations is looking at archaeology. So archaeology gave us a lot of information regarding our history as a species. So we all belong to the Homo sapiens species, which is the representative species of the human genus, of mankind, of humanity. So the Homo sapiens species, it emerged in Africa around 200,000 years ago. And from Africa, they migrated to all the regions of the planet. First to the Arabian Peninsula around 100,000 years ago, and after that, they moved to different directions. Around 50,000 years ago, the first human groups, they reached South Asia and Oceania. Around the same time, they went to Europe. And 20,000 years ago, some ancient human groups, they moved to North Asia or Northeast Asia. So in the meantime, while they were moving around these different continents, these different regions, they met with other kinds of humans. So as I told you, nowadays, the Homo sapiens is the representative species of humanity. But in the past, there were different kinds of humans. And one of those kinds are the Neanderthal. So the Neanderthals were mostly found in Europe, and they lived in small groups. As Homo sapiens, they also produced stone tools, so they shared some cultural traits with the Homo sapiens. So other kinds of humans are the Denisovans. So Denisovans mostly lived in Asia. But little is known about the society and culture, mainly because we don't have many remains of the Denisovans. We only have some fragments of bones and also some spare teeth. So all the information we know from Denisovans come from these fragments and these spare teeth. So both Neanderthals and Denisovans, they disappeared around 40,000 years ago. So before disappearing, they interacted with Homo sapiens. And we know that this relationship with the Homo sapiens were not exactly a conflicting interaction. They didn't disappear because of warfare or because of conflicts, but probably because they were assimilated by the Homo sapiens. And I will present, further in my presentation, I will present some evidence of this disappearance by assimilation. So after 20,000 years ago, we have the first migrations into the Americas. So around 20,000 years ago, during the last glacial maximum, that means the last ice age, there was a massive land bridge in the Bering Strait. So the Bering Strait is this region between Northeast Asia and Alaska. So during the last ice age, the ocean waters, they were trapped in continental ice sheets. So the overall ocean level was lower than today, and this allowed for the emergence of a massive land bridge between linking Northeast Asia and the Alaska. So this was the main entry point of ancient human groups into the Americas. However, when they reached North America, things started to get confusing. If we look at the archaeological records found in North America and in South America, regardless of the region that you are looking at the Americas, they show very similar ages, around 15 to 13,000 years ago. So archaeologists started proposing new hypotheses to explain why we have these similar ages regardless of the region that we are looking at. So one of the hypotheses proposed was that some human groups, they came from Europe to Eastern North America, and that other human groups, they came in a trans-Pacific route from Oceania to South and South America. However, these migrations, they generate even more questions. For example, how would ancient human groups be able to cross the whole Pacific from Australia directly to South and South America? So it was clear that at this point that archaeology alone could not provide the necessary details that we needed to understand the whole settlement of the Americas. And that's when the researchers, they decided to turn to analysis of ancient DNA. So the analysis of ancient DNA is another way to investigate the details, the main aspects of the settlement of the humans of the past. So ancient DNA can be found in ancient human remains. So in this case, I show a picture here of a human bone, but also human teeth and hair. And they can also and ancient DNA can also be found in archaeological sediments and soil samples from archaeological sites. So nowadays, ancient DNA, the study of ancient DNA is an established scientific field. And last year, this field was recognized by a Nobel Prize in medicine that was awarded to the Swedish researcher Svante Pääbo. Svante Pääbo is one of the pioneers of this field, and he is also the principal investigator of efforts to sequence and analyze the DNA found in Neanderthal and Denisovan remains. So he is responsible for analyzing DNA found, the DNA of archaic hominids, the extinct hominids, the ancient hominids that I presented you in my second slide. So, but let me talk a little bit about DNA. So DNA is present in every organism and is a long molecule composed of many copies of 4 other smaller molecules. They are called the nucleotides, specifically adenine, cytosine, guanine, and thymine. So these nucleotides, they form the DNA. They appear in many copies throughout the DNA. DNA, in turn, is the main material that the chromosomes are made up, and the chromosomes can be found in the cell nucleus. So each cell of our body carries a copy of our whole genome. So all the DNA inside a cell is known as a genome, and each cell in our body carries one copy of our genome. There are approximately 3 billion nucleotides in the human genome. So this is a really big molecule. And how do we analyze this big molecule? So DNA is analyzed by comparison. When we compare two human genomes, we found in average only one difference in 1,000 nucleotides. So 1 in 1,000 nucleotides is different. So this difference is called the variant or mutation or polymorphism. So these differences are used by us in our analysis to infer ancestry. And based on the different ancestors that we find, we can propose migrations between continents, between populations. But to find, however, to find these differences in the genome, given that the genome is a really big molecule, we are talking about big data, we need powerful computational tools to identify these differences in the genome. And that's why we are doing this analysis here in the Department of Electrical Engineering and Computer Science. So hundreds of ancient genomes have been obtained in the Americas. So these ancient genomes were published between 2010 and 2018 in 12 ancient DNA studies. So as you can see here, most of these genomes were associated with archaeological sites in the USA and Canada. And until 2022, so until we started doing this research, these hundreds of ancient genomes of the Americas, they had never been analyzed as a single data set. Although this is true, although they had never been analyzed as a single data set, these 12 distinct ancient DNA studies, they provided us very important information that I summarized here in this figure. So this figure tells us some of the steps that the first human groups that reached Americas took to settle the continent. So first, after they crossed the Beringia, some human groups, they settled around the Alaska, and they gave rise to the ancient Beringian ancestry. This ancient Beringian ancestry is representative of one archaeological site in Alaska called the Upward Sun River. After that, some other groups, they moved further into the continents, and then two major genomic ancestry appeared in the Americas. One of those major genomic ancestries is called the NNA, or Northern Native Americans, and the other major branch is the SNA, or Southern Native Americans. So descendants of these Southern Native Americans, they moved all the way down to South America, reaching even Tierra del Fuego in the extreme south of South America. So nowadays, most of the present-day populations of the Americas, they belong to this SNA branch. So I will talk more about some information presenting this figure. For example, I will talk more about the ancient Beringian ancestry and the Upward Sun River archaeological site, the USR archaeological site. And in South America, I will talk more about the Lagoa Santa archaeological site, from which Somejoro 5 was found, and it's located in Southeast Brazil. So further in my presentation, I will talk more about this information. So we decided to analyze all these individuals that were found in the Americas, all these genomes associated with the archaeological sites in the Americas, as a single data set. We knew that given that they were studied in different studies in different worlds, we knew that there were still some relationships, some genomic affinities that were not reported. So by analyzing these individuals together, we knew that we could still go further in this analysis, in the analysis of ancient DNA, in the analysis of migrations in the Americas. So let me show you one of the analysis that we performed. I know that this figure is very confusing to you, so I will try to explain this figure by my best. So here we analyze hundreds of ancient genomes from the Americas, plus 278 present-day individuals from the Simons Genome Diversity Project, SDDP. So SDDP is one of the most important genomic projects ever done, because the researchers behind this project, they aimed to sequence native, aboriginal, and indigenous peoples of the different continents of the world. So here we are comparing our ancient Native American genomes with present-day genomes from indigenous, native, and aboriginal peoples of the world. So here in this figure, each vertical bar here represents a single sample, and in the top two rows, we can see the ancient samples of the Americas, and in the bottom row, we can see the the present samples from different continents of the world. So, and also each color here represents a different ancestry. So by doing this analysis, we wanted to see first, how many ancestries can we find in this data set? And second, how these ancestries are related to each other, how these populations are related to each other. So each ancestry in this data set is represented by a different color. So for example, if we look at the present day individuals of the Americas, we can see that the green color, the green ancestry, is the predominant ancestry in the Americas. Outside of the Americas, we also see this green ancestry in Central Asia and Siberia. So this is an evidence that indeed the first human groups that settled the Americas, they had some like genomic relationships with the populations from Asia. And when we look at the ancient individuals, we also see the green ancestry, the green component as the predominant genomic component in the ancient individuals. But also we see all the other colors as well, the dark blue, the light blue, the red colors. And this is showing us that in ancient South Americas, there was a higher diversity than in present day Americas. This is probably an outcome like this decrease of diversity. So nowadays that we can see that in present day Americas, these individuals are mostly homogeneous. So this decrease of diversity is probably associated with the European colonization of the Americas. Okay, so we decided to move on studying the diversity of the ancient individuals, and we find a higher diversity in ancient North America in related to ancient South America. So what we found here, like in this plot, so each data point represents a different individual, and this green area, this gray area in the background represents the density of these individuals. So we see a higher density in North America when we are looking at values around 0.4 and 0.5 of overall diversity. Whereas in South America, we see a higher density of individuals around 0.1. So overall, the diversity in North America is higher than in South America. So this is also consistent with the hypothesis that Beringia, northern North America, was the main entry point of ancient human groups into the Americas. And why this is an evidence? Because if North America is receiving new ancestries coming from Asia, that's gonna keep the overall level of diversity at higher values. Whereas in South America, we only see lower values. That means that no major migration outside of the Americas came directly to South America. When we look at the countries, we see a similar pattern that USA here in the right and Canada in the middle, they show the highest levels of diversity in the Americas, whereas most of the countries in South America, for example, Peru, Chile, Belize, and Argentina, they show very low levels of diversity. But there is an exception to this rule, which is ancient Brazil. So ancient Brazil shows a high level of diversity similar to Canada and USA. So this is a very intriguing finding. And later on in my presentation, I will give you some evidence of what may have happened here that kept the overall level of diversity in ancient Brazil at higher levels. So here we decided to move on with different analyses. And the next analysis that we did was an analysis of genomic affinity. So here in this plot, every individual are represented by a data point. And we wanted to see how they related to each other in a two-dimensional space. Also, every different population is represented by a different color here. So the present-day individuals from the Americas, they are represented by the blue color. The ancient individuals of the Americas, they are represented by the grayscale here. And what we can see is that most of the ancient individuals of the Americas, they fall closer to the present-day individuals of the Americas. But there are also exceptions to this finding. And we find a few individuals, a few ancient individuals falling closer to populations from Central Asia and Siberia. And let's zoom in this region of the graph so we can see which individuals are those. So one of those individuals is the USR1. So USR1 was found in an archaeological site that I already talked about, the Upward Sun River archaeological site in the Alaska. We knew before this analysis that USR1 was the representative of the ancient Beringian ancestry, but we thought USR1 was the sole representative. He was the single representative of the ancient Beringian ancestry. But during this analysis, we found that all the two individuals share a very similar ancestry. Actually, they belong to the ancient Beringian ancestry. So let's see where these other individuals are located in the map. So here I have a slice of North America, and we also can see some portions of Northeast Asia. And here we see the locations of all three individuals. So USR1, we already know it was found in the Alaska, but I-0719 was found in the Aleutian Islands. So the Aleutian Islands are part of the Beringia. Also, we have the Sakak individual. So the Sakak individual was found in an archaeological site in Greenland, and this was totally unexpected to us. So based on the chronologies of these individuals, based on the ages of these samples, we can say that the USR1 is the oldest sample of those three. So we can say that the ancient Beringian ancestry moved from the Beringia itself, and some of those descendants of some of the groups that belonged to the ancient Beringians, they moved to Greenland, eventually settling in Greenland, and Sakak was one of their descendants. So let's go back to our map, summarizing the main findings of the Americas. Now we can update this map. Now we know that the ancient Beringian wasn't restricted to the Alaska, but also they moved all the way to Greenland. Still, looking at this map, we know there are still large portions of the Americas that remain unexplored by ancient DNA studies. Some of those are Central USA, Southern USA, also Central South America, and I will highlight a different region, which is the Northeast Brazil. So why am I highlighting this region? So Northeast Brazil houses one of the richest archaeological areas in the Americas, called Serra da Capivara National Park. Also, this region is very important to me because I was born and raised in the city of Fortaleza, in this region, Northeast Brazil. But let's talk about Serra da Capivara National Park. Why is this important? So Serra da Capivara National Park houses thousands of archaeological sites. It's a massive archaeological area. So there are thousands of archaeological sites in this area. Each site has dozens of cave paintings. Also, there were hundreds of thousands of archaeological artifacts found in this area. And given the richness of this area, since 1991, this site was designated World Heritage Site. So Serra da Capivara is also believed to be the source of all the prehistoric occupations in Northeast Brazil. So especially when we are looking at Eastern Northeast Brazil, we find cave paintings that are very similar to cave paintings found in Serra da Capivara. In this case, I brought the example of the back-to-back motif. So this motif is found in many archaeological sites in Eastern Northeast Brazil, and it is believed to be originated in Serra da Capivara. So given that we find the same motifs of cave paintings in different archaeological sites, we believe that there were many migrations that happened in the past in Northeast Brazil. So we decided to study two new samples from Eastern Northeast Brazil. These samples are called Brazil 2 and Brazil 12. So the idea is that analyzing the genomes from these samples would allow us to understand the migrations that happened in the past in Northeast Brazil. And we did this by comparing these two ancient samples from Northeast Brazil with previously published samples, ancient samples from the Americas. And I can talk to you about some of these samples. For example, N-1 that was found in Montana, Spirit Cave found in archaeological site in Nevada. These are one of the oldest samples found in the Americas. Also, we analyzed PAPV-173 from Panama. This is the single sample from Central America that we analyzed in this work. Other three samples collectively known as the Ilave, they were found in Peru. So Ilave is a region from Peru. So we believe that these samples, they belong to ancient peoples that occupied the Peruvian highlands, the Andes. Also, one sample from Tierra del Fuego from Chile, sample A460. Two samples from Uruguay, CH19B and CH13. One sample from Southeast Brazil, Sumidoro 5, that I already talked about a little bit about the sample. Sumidoro 5 was found in Southeast Brazil. And we also compared our samples to the SDDP. So I already presented SDDP to you. So it's that project in which native, aboriginal, and indigenous peoples were sequenced. So the idea here is to analyze our ancient Native Americans to present-day Native Americans. And here is our first result of this analysis. So here's the first result of this analysis. This is an admixture analysis. I already presented a result to you that is similar to this. So here again, every vertical bar represents a different individual, and every color represents a different ancestry. And I want you to focus on the right-hand side of this figure, in which we have this orange ancestry. We found that this orange ancestry can only be found, is restricted to three samples: CH19B, Brazil 2 from Northeast Brazil, and Sumidoro 5 from Southeast Brazil. So no other sample in our data set carried this orange ancestry. And this probably explains why we see a higher diversity in Brazil, because in Brazil there was a distinct ancestry that could not be found anywhere else in the world. And that's why we have a high level of diversity compared to other samples from other countries in South America. We also found that this distinct ancestry is located along the Atlantic coast, was found along the Atlantic coast. So CH19B was found here in Uruguay, Sumidoro 5 found in Southeast Brazil, Brazil 2 found in Northeast Brazil. So these three samples, they carry that orange ancestry. So this ancestry was restricted to South America's Atlantic coast. We did a different analysis this time. This is called a tree mix analysis, but although this is a different analysis, it returned a very similar result to our previous analysis. So you can think, the tree mix analysis returned a tree, and you can think about this tree as a genealogical tree in which we have the ancestors and the descendants. So in this case, the ancestors, they would be located in the top left portion of the graph, whereas the descendants would be located in the bottom right portion of the graph. And also this result, it basically recapitulates, it mirrors the migrations that I showed you at my second slide, in which we have an African population closer to the root of this tree. So, and then after the African migrations, what we had migrations toward Oceania. So the second populations that we see here in this graph is an oceanic population, the Papuans. After the Oceania, we see the populations from Europe, English and French. After that, we see populations from East Asia, Han and Japanese. But when we get to the Americas, we see two major branches in this tree. So one of the branches included samples found closer to the Pacific coast. For example, N-1, Spirit Cave, Ilave from Peru, and A460 from Chile, whereas the other branch included the individuals that were found closer to the Atlantic coast, C819b, Somidoro 5, Brazil 2, and PAPV 173 from Panama. So we found very similar results in this analysis compared to the other analysis that we did. But this analysis also gives another information, which is who is the ancestor of who. In this case, we can say that Somidoro 5 in this Atlantic coast branch was associated with the ancestors of Brazil 2. In turn, Brazil 2 was associated with the ancestors of both C819b from Uruguay and PAPV 173 from Panama. So what this graph is telling us is that Somidoro 5 was probably the ancestor of Brazil 2 in northeast Brazil. So apparently we are seeing here a migration that started in southern Brazil and moved north to northeast Brazil. And from northeast Brazil, it moved even more north to Panama. So this is the first time that we get an evidence of a south to north migration. All the migrations that I showed you here today are from the opposite direction, from north to south. But here we are finding evidence of something that we never saw, something that was never seen, which is a migration from south to north in the Americas. So we decided to expand this analysis, the tree-mix analysis. So we included more populations, more samples from the SDGP, from present-day native, aboriginal, and indigenous peoples, and also we asked the software, the tree-mix software, to show us the most probable migrations that involved these samples, this data set. And here is the result. So this is very confusing, but I want you to focus on this migration vector here, this migration edge, in which it starts in C819b and PAPV 173. So the software suggests a migration that started in Uruguay and moved all the way to Panama. And this, as I told you, this was totally unexpected to us. We never saw any evidence of a north to south, a south to north migration, sorry. And here we are finding decisive genomic evidence that these migrations also occur in the Americas. So let me show you a figure in which I summarize all the main findings of our research. So here, I present two big migrations that occurred in South America in the past. So the first migration, represented by this yellow arrow, represents the main migration that came from North America to South America. So this migration occurred along the Atlantic coast or closer to the to the Pacific coast or closer to the Pacific coast, passing through Central America, Ilave in the Peruvian highlands, all the way down to South and South America, in which we found this individual A460. So at some point during this big migration, during this continental migration, some groups, they branched out. They moved from the Pacific coast, from the west coast of South America to the east coast of South America, eventually settling around Lagoa Santa. Lagoa Santa is the archaeological site of Somidoro 5. And once they settled this archaeological site, this Lagoa Santa archaeological site closer to the Atlantic coast, they moved both north towards northeast Brazil and Panama, and south towards Uruguay. So these are the main findings of our work. And here is the last slide that I talked to you about migrations in this presentation. But besides these results, we also found other intriguing results that I'm going to present in this next slide. So I already talked to you about the Swedish researcher Svante Pääbo, who was awarded the Nobel Prize in Medicine last year. So I told you that he was the principal investigator of analyzing ancient the DNA, the ancient genomes from Neanderthals and Denisovans. And one of the conclusions of his research is that every population, every present-day population, or almost every present-day population on Earth, they carry some fragments of Neanderthal and Denisovan DNA in their genome. That means that all of us who are alive today, we carry some fragments, some small proportions of the Neanderthal and the Denisovan DNA in our genomes. So usually we have more Neanderthal than Denisovan genome in our genomes. So this is already known, but we wanted to know if any of the ancient individuals of the America, they also carried these fragments. And we tested 10 individuals from South America and Panama, and we found that all of them carried at least some fragments of the Neanderthal genome. What does it mean? Does it mean that the Neanderthals and Denisovans, they came to the Americas? No, it means that during that time that the Homo sapiens interacted with the Neanderthals and the Denisovans, these interactions probably generated offsprings. And these offsprings, they carried both Homo sapiens, Neanderthals, and Denisovan genomes. And the descendants of these offsprings, they came to the Americas and eventually settled the continents. So we knew that we could find some fragments of this archaic ancestry in our ancient individuals based on the findings on the present-day populations, but we didn't know that we could find even more than that. So we found that these two individuals, one from Panama, Ph.D. 173, and CH19B from Uruguay, they carried more Denisovan than Neanderthal fragments of DNA. So this was totally unexpected as well. We were expecting to see more Neanderthal than Denisovan, but this is the first time in the world that we are reporting more Denisovan than Neanderthal in ancient individuals. I already talked to you. So before saying this, I already talked to you that there was a special relationship, a distinct genomic relationship between CH19B in Uruguay and PPV 173 from Panama. But this archaic ancestry is something else. This is totally unexpected. We didn't expect to see this signal of more Denisovan than Neanderthal in the Americas or in any place at all. So we still don't know exactly how this signal appears in the Americas. So this is a mystery to us. We plan to expand our research, this test, to other samples in the Americas, to other samples outside of the Americas. We want to see what is the extent of this signal in the world. And based on this, probably we will have a better idea, a better explanation on how this signal came to the Americas and what is the extent of this signal in the Americas and in the world. But for now, we don't have this information yet. These are next steps that we are going to develop in the future. So yeah, this is the last main result of our research. But before finish my presentation, I have to talk about three people. One of them is my current postdoc advisor, Dr. Michael DeGiorgio. He was responsible for many of the analyses that we did here. Even some of the results that I'm presenting here, he was the person who did this analysis. So yeah, I cannot talk about this project without talking about his name. Most of the insights that we have in this research, they came after lengthy discussions between us. A second very important person to this research is Dr. John Lindo, who is a professor, a faculty at Emory University. He was the person responsible for sequencing the ancient genomes for Northeast Brazil. So if we have this data, if we can analyze this data, it's because he offered to sequence these samples from Northeast Brazil. Last but not least, I'm going to talk about Omer Gottman. He's an assistant professor, he's associate professor at SUNY Buffalo, University at Buffalo. He was my PhD co-advisor, and many of the insights that I had during the beginning of this research, was due to our discussions as well. So he gave me valuable insights that I was able to apply in this research. And some of the analysis was also done because he allowed the right environment to do this analysis, at least the initial analysis that we did. So I cannot talk about this research, and I don't want to give you the idea that this is only my research. So I cannot talk about this research without mentioning these three people. Okay, so, and that's it, I believe. So thank you for your attendance. Thank you for your attention. Thank you for your patience. I will gladly answer any questions that you might have right now. But if you want, please feel free to contact me. If you have any questions after this presentation, you can send me an email, and I will keep my email here so you can see this and send me anyway any questions that you might have. So thank you so much.
Thank you so much, Andre. That was really fascinating, because we all make assumptions about where you come from and how that's figured out. I welcome anybody in the audience that would like to put some questions into the Q&A feature. Happy to address them. But I actually have a couple for you right now.
Okay.
So the first one is, to talk about how do you actually extract the DNA to analyze? And do you yourself, or do you do that at FAU, or does somebody else do that?
Right. Yeah, thanks, Jean. This is a very important question. So unfortunately, we don't do the extraction of DNA here at FAU yet. Maybe in the future we will be able to do this, but right now we count on a huge collaborator, which is Professor John Lindo from Emory University. He has all the structure to extract ancient DNA. When we are extracting ancient DNA, we have to be very careful to avoid contamination from present-day samples, even from us. So he has a specific structure, he has specific equipment to extract DNA. And once he gets the DNA, he also performs the sequencing of this DNA. And after the sequencing, he shares all the files that he has regarding this DNA to us, and we can do all the computational analysis here at FAU. So all of these analyses that are presented today here were done here at FAU. All the computational analyses are done here at FAU, but the extraction is done elsewhere.
See, okay. So another question that just came in is, do you have Brazilian partners in your research?
So the Brazilian partners in my research are my previous Ph.D. advisor. So part of this research, it was started during my Ph.D. So he also provided a lot of a bunch of very important information regarding the archaeological context of those samples. So my advisor had this important role of characterizing the archaeological context of this sample. So we also could not have done this without his help. He was also responsible for getting these samples and send them to the US so we could sequence the samples, and then after that we could analyze the samples. So also my PhD advisor, who is also from Brazil, he played an important role in this research as well.
So earlier there was a question too, and this might actually go to what you just said, since you're from Brazil, your PhD advisor was from Brazil, and so a lot of the work that you were doing had very unique properties in the sense of a particular archaeological finds.
Right.
Has the type of research that you've done, have you done that in other geographic locations, or is it being done in other locations?
Yeah. So right now we are analyzing different populations, not only in South America but also North America, and we are always analyzing ancient genomes from different parts of the planet. So, for example, we keep still analyzing the Neanderthal genomes, the Denisova genomes, to see what we can find in our samples as well. And these genomes, the Neanderthal and the Denisova, they come from Eurasia, they come from Europe and Asia. So in that sense, we are always analyzing samples from different parts of the world. And right now we are analyzing other samples from different native populations of the Americas as well, not only from South America, but also in North America.
And that's interesting. So another question that came up
Mhm.
then is when you see overlapping characteristics of genome of two samples from different sites, how do
Okay.
you determine the migration route that is going from one to
Mhm.
the other or the other way around?
So these migration directions are basically decided using the similarities and differences in these genomes. So when that's why we also include samples from present day populations. So when these similarities or differences are more close to other populations outside of the Americas. So let's say that there is a variant, a mutation that is similar to a mutation that we see in Africa. We consider this mutation to be the ancestral state of the mutation. But if we see a mutation that is similar to populations in the present-day populations of the America, we consider this mutation the descendant, the alternate state of the mutation. So based on similarities with other populations, let's say from Africa, from Oceania, from Asia, we can say that this mutation seems ancestral. And then we can propose a direction between this mutation and other mutations that we might find in the data set.
Okay, great.
Yeah,
And so that has a follow-up question based on some
Mhm.
of the other locations in geography.
Okay.
Rachel Kaur said, thank you for this presentation. I read that some South Americans have DNA that is found in Polynesian societies,
Right,
that this does not indicate migration from this area.
right.
What's the explanation for this, and why is it not found in other Native American populations, or is
Mhm.
this part of the mystery you were discussing?
Right, right. This is a big mystery yet. So this is a very important question as well. I presented to you that in the ancient samples of South America, we found some Neanderthal and Denisova fragments. But these findings doesn't mean that the Neanderthals and Denisovas they came to the Americas. It means that their descendants mixed with Homo sapiens, and then these offspring came, or the descendants of these offspring came to the Americas. So it's the same for the Polynesian ancestry. Although we find this signal in South America, we still don't have any decisive archaeological or genomic evidence that this signal came directly from Oceania to South America in a cross-Pacific, in a trans-Pacific route. We can say that individuals that carry this signal, they came from Asia or Northeast Asia, they crossed the Beringia, and they settled in the Americas. So we still don't have any evidence, be it material, be it genomic evidence that they came directly across the Pacific, but rather the descendants of these people carrying the fragments of Polynesia or Oceanic DNA, they came to the Americas through the Beringia.
I see.
Yeah.
So I actually am going to pivot it to another question,
Okay.
and it has to do with data sets such as All of Us, the NIH All of Us genomic sets and everything. How does the research that you're doing interact at all, if at all, with data sets like this?
So usually we will use these data sets as reference panels to identify different ancestries. So basically we look at present day diversity to understand the past diversity. But there are other aspects that we can also infer from present day data, and we can also apply these inferences to our ancient data sets. And these aspects are, for example, adaptation to different environments, some possible diseases that occur in the present that might have occurred also in the past. So by studying the mutations, the variants, the polymorphisms in present day populations, we can have a better idea of the environment that the ancient individuals faced as well. For example, the environments involving diseases, the environments also that involved very different like temperatures, different levels of oxygen, of hypoxia or not, or something like this. So we use the modern day populations to infer the lives of ancient populations to see which obstacles they had to overcome, to
Mm.
see which barriers, which diseases they also had to overcome in the past. So basically, this we compare these populations, the present day and the ancient, to better understand our past.
Oh, very interesting. Very
Yeah,
good.
thank you.
And then because of the data that's there, does that inform other, not necessarily public data sets, but for instance, so there's the All of Us, which is the data set at NIH, but what about for profit? So things like Ancestry.com and things like 23andMe. Does this data or information inform any of those platforms?
So unfortunately, we don't have access to these kinds of data. We would love to have it. I'm sure there are many information that we could achieve with these data sets, many conclusions that we could achieve with data sets, but right now we are only working with public data set. And we can only provide conclusions involving this public data set. But maybe in the future we will have access to other data sets, and that would be great, definitely.
Maybe we'll put that out there for some companies to listen to.
Right.
Exactly.
Yeah.
So there's one more question,
Okay.
and also a comment. Roberto Santos said, congratulations to you and excellent job for the presentation. And then the final question is, do you know of any researchers who are studying the Homo sapien presence and migrations in the Americas?
So there are many groups who study the Homo sapiens in the Americas. Especially here in the US, we have the Dr. Dave Dry. He's the principal investigator of a lab in Harvard University. So he also studied the settlement of the Americas in the past. In Europe, we have two major centers. One is the Max Planck for Evolutionary Anthropology in Germany, the Max Planck Institute in Germany, which is headed by Svante Pääbo that I presented here. The other one is in Denmark, in Copenhagen. There is the Geogenetic Center at the University of Copenhagen, which is also one of the major groups in our field. So, but there are also other labs in different parts of the world. Even I believe that in Asia, in Oceania, there are other labs who also study the settlement of the Americas using ancient DNA.
Great. And with that, we'll wrap it up. And I want to thank you again, Andre, for coming on and
Thank you.
Research in Action and to our audience. And I appreciate it. And you all have a great day. Thank you so much.
Bye-bye.
Bye-bye.